So now let's talk a little bit about our blood vessels. So now we're getting into actual exercise 3 physiology. We finished up with the last lecture talking in a little bit more detail about the heart and cardiac output. Now we're coming into the topics associated with the blood vessels as it relates to their physiology. So first, let's talk about the different types of blood vessels. Blood vessels are categorized in 3 general groups. You have your arteries, which we see here, which are vessels that carry blood away from the heart. And as you'll see shortly, we have the opposite of that, which would be veins and those vessels that carry blood towards the heart. And finally, where the two meet, we have our capillaries, and our capillaries are going to be where we undergo capillary exchange, or the process of dropping off the good and picking up the bad. So let's focus here a little bit on our arteries. Arteries are defined as carrying blood away from the heart. They typically have a very rapid transport. And this is because with the fact that they're carrying the blood away from the heart, they're going to be exposed to the highest pressure and the highest force. With that high pressure and high force, we see arteries tend to be much thicker in comparison to your veins, as you saw will see when you look at them under the microscope. And they have a much thicker tunica externa, as well as this guy here, which is the tunica media. 3 layers of your blood vessels, which you're going to see for all blood vessels with the exception of capillaries, is the tunica interna, the tunica media, and the tunica externa. The tunica interna, which is predominantly made up of endothelium and a basement membrane with a little bit of elasticity, is the innermost layer of your blood vessels, and this is what the blood is actually rubbing up against. In the middle, we have the tunica media, which is made up of smooth muscle. The thickness of this smooth muscle will vary depending on what artery we're talking about. But this smooth muscle here is for vasoconstriction and vasodilation, which is going to have a variety of functions, from controlling the distribution of blood flow to adjusting blood pressure. Finally, we have the tunica externa, which is also sometimes referred to as the tunica adventitia. This is going to be predominantly areolar connective tissue to anchor and kind of glue, so to speak, the blood vessel into its surrounding tissue. By the way, the tunica interna is also sometimes referred to as the tunica intima. You may have seen that. Now, as I said, arteries require a rapid transport. Larger arteries closer to the heart often serve as pressure reservoirs, as they have a greater amount of elasticity, so they can absorb that pressure and then recoil to continue it on. Arteries have a much larger radii or diameter for increased flow, as they distribute the blood coming from the heart to the different regions of the body. Now, as you'll see shortly, as arteries get progressively smaller the further away they get from the heart, they turn from arteries into arterioles, which again have all 3 same layers, but they're much, much thinner. Now, here's a quick question for you. One of the common things or misconceptions that students get comes to this question here. What type of blood flows in arteries? Is it oxygenated or deoxygenated? Think about that for a minute and come up with your answer. Well, hopefully you are saying both. Remember, arteries are defined as those vessels that carry blood away from the heart. Now, if we're carrying blood away from the left ventricle of the heart, then we're carrying oxygenated blood through systemic arteries. But if we're arteries and we're leaving the right side of the heart, well, then we're going to the lungs, which means we're going to the lungs to get oxygen, which means our pulmonary arteries would actually be carrying deoxygenated blood. Okay. This is also why we can have arteries that are red and arteries that are blue, because systemic arteries, since they carry oxygenated blood, will appear red, whereas pulmonary arteries, since they carry deoxygenated blood, will appear blue. And this is an important note, because a lot of students get caught thinking arteries red, veins blue, and that is not accurate, not true at all. So continuing on with our discussion of vessels, remember what I had said, as arteries get progressively smaller, they turn into arterioles. Arterioles are the final distribution point before we give that blood up to the capillaries. Arterioles have a smaller radii, are responsible for distributing the blood to all the local tissues, and still possess all three layers, the tunica interna, the tunica media, and the tunica externa, but they're much, much thinner. But being that they have a smaller radii, you don't necessarily need as much smooth muscle. Okay? Now, remember what I had said, that your arteries and arterioles with that smooth muscle layer are going to be responsible for vasoconstriction and vasodilation. Now, when that vasoconstriction and vasodilation occurs at the artery level, we're mainly controlling or regulating the distribution of blood. But what we see at the arterial level, this is where we're going to regulate vascular tone and vasoconstrict and vasodilate for the regulation of blood pressure. A little abbreviation here. To regulate blood pressure, primarily by regulating the concept of TPR, which we're going to talk about here in a couple of slides. So kind of hold that thought as far as wondering what TPR is. As your arterioles eventually get smaller and smaller, they lose all of the tunica media and really most of the tunica externa to really just become the tunica intima. And when that happens, they turn into capillaries. Capillaries is a site of exchange between the blood and the tissues in which they're running through. So this is where we're going to drop off the good and pick up the bad, for the most part. Of course, in the lungs, we're picking up the good and dropping off the bad, but that's a different story. Now, at the point of the capillary levels, these guys are very small in diameter, and they're so narrow to the point where they really only allow a single blood cell through at a time. And this is, of course, to maximize exchange. If you think about it, if drive-thrus allowed 5 cars through at once, something's going to get missed. Okay? As those capillaries leave the tissues, they eventually come back together to form venules and finally veins. Again, these guys defined by carrying blood back to the heart. Again, we return to having all three layers, your intima, media, and externa, but they're, as you can see, much thinner in comparison. And since we have much less pressure in these areas, you don't need the same amount of thickness. One of the, in addition to being thinner, they also have something unique to veins, which are these valves. These valves are very important in preventing backflow, because in many cases, for 2/3 of our body to 3/4 of our body, we have to work against gravity to get the blood back to the heart. We need some different ways to help us do that. And one of the ways we can do that is by using valves. These valves prevent backflow so that if we do get the blood moving in the right direction, it doesn't just flow right back down again due to gravity. So how do we get the blood back to the heart in reference to gravity? How do we make the blood return? Well, this whole idea of returning blood to the heart, or this concept of venous return, which we had mentioned previously, is aided by 3 different mechanisms or concepts, one of which is those valves, which we already talked about. We use these valves to prevent backflow, but backflow doesn't necessarily mean that we're making the blood go up. So how do we make the blood return to the heart? Well, we use 2 pumps. First, we have muscular pumps. What you're going to see, particularly if you pay attention in class, is that your veins are strategically placed between muscle groups. What this means is when these muscle groups contract, they squeeze that vein, and if you squeeze the vein, blood is forced upwards. Of course, as soon as you stop squeezing the vein, the blood's going to want to go back down again, but we stop that with the help of those lovely valves. This is why when we have a bedridden individual, we often will go in there and forcefully move those limbs and contract those muscles to aid in circulation and prevent the blood from pooling and clotting. Now, the problem is muscle pumps only work in your extremities, in your arms and legs. So what happens when we get to the abdomen? Well, in the abdomen, we use your respiratory pumps. If you guys recall, either from lectures or your readings, blood flows according to a pressure gradient. So blood will always flow from an area of high pressure to low pressure. Well, what we see here is when we squeeze those veins, we were forcefully increasing the pressure. And that's what made the blood move upward. But when we get to the abdominal pelvic cavity, we don't have a bunch of skeletal muscles flowing in there or found in there. So we can't use that squeezing action to increase the pressure. So what are our options? Well, if we need to get blood to flow from the abdomen back to the thoracic cavity, we really only have two. Is to somehow make the pressure in the abdominal pelvic cavity higher, or in a different manner, make the pressure in the thoracic cavity lower. And that's actually what we do. Whenever we inhale drops the pressure of the thoracic cavity. And that drop in pressure from the thoracic cavity, which, as far as the respiratory system is concerned, is used to suck air in, actually has a secondary function with the cardiovascular system to suck air up. So we can suck the air up, suck the blood from our abdominal veins into the thoracic cavity and back to the heart. And that's referred to as your respiratory pump, or thoracic draw is another term that's often used for that. So again, let's take a minute to see if you guys have been following along. I'm going to leave it on the slide for a second while you read these questions and see if you can answer them. If you cannot answer them, then you're going to want to go back and review what we just talked about. And if you're still confused, email me or come see me so we can clarify and get you on the right track to success. Well, finally, let's talk about how we direct the blood to flow to different areas. The idea here with factors affecting blood flow is this idea that different tissues need different amounts of blood. When you're running a marathon, you're going to need a lot more blood flowing to your legs than you would if you're sitting on the couch. So how do we direct blood flow? How do we control where it goes? Well, we control blood flow via two factors. First of all, we got to give reason. We have to give blood a reason to flow. And that reason to flow is, of course, the contraction of the heart and the blood pressure that's created when the heart contracts. Now, here's the thing. You could create all the pressure in the world. Does that mean blood's going to flow? You could put all the pressure of the world on with your bike, but if your bike's stuck in mud, it ain't going to go anywhere. And that's because there's too much resistance to the movement of that bike. That bike is stuck in the mud. So we have to take into account the fact that blood doesn't just flow in a vacuum. Okay, it's got to flow through tubes, and those tubes create resistance. So we also have to take into account the concept of resistance. And the idea is that we've got to create enough force through cardiac output and a high enough blood pressure to overcome the resistance of those vessels. So both of these very important to remember. And if one will always or ultimately affect the other. Now, we talked quite a bit about blood pressure and cardiac output in the earlier sections of this lecture, so now we're going to talk about the concept of resistance. Because again, anytime you start something moving, it will eventually slow down. Right? This is why you got to keep pedaling your bike. This is why you got to keep putting your foot on the gas, because there's resistance as your car travels across the road, or your bike pedals go across the gravel. And that resistance slowly eats away at your force. Resistance itself is referred to, or occurs with friction. Friction occurs when two things rub against one another. In our car analogy, when the tires rub against the pavement, when the car pushes against the air, all of that creates resistance, which eats away at the force, causing you to have to push on that gas pedal again. But in the case of our blood, the primary resistance factor originates when the blood rubs up against the vessel walls. So anything that makes that occur more or to a greater degree is going to increase resistance. Now, your book talks about a variety of or your manual talks about a variety of resistance factors. But three of the most important resistance factors are going to relate back to the average diameter of your blood vessels, how thick your blood is, and then your total blood vessel length. What we're going to see is the smaller the diameter of your blood vessel, the greater the resistance. The idea is in a small diameter blood vessel, more of that blood, overall, more of the blood is rubbing up against that vessel wall, leading to higher friction and higher resistance. Whereas in a larger diameter vessel, most of the blood is in the lumen, not touching the vessel wall, which means you have lower resistance. Blood viscosity is the thickness. Think about sucking up a Coke versus sucking up a milkshake through a straw. It's much easier to drink that Coke than it is that milkshake because it's thicker, and thicker blood creates more friction. Finally, total blood vessel length. In this case, it's not that more blood is rubbing up against the vessel wall, it's that there's more vessel for the blood to rub against. This is why individuals that are obese and have extra blood vessels as a result of all the excess tissue, adipose tissue spread throughout their body, tend to have high blood pressure because their heart has to create a lot more force in order to distribute blood through all of those extra vessels. Now, what we end up seeing is if we take into account all of these resistance factors, the three main ones, as well as additional factors that your less important factors that your book mentions, what we end up having is this concept of total vascular resistance, or excuse me, systemic vascular resistance, or total peripheral resistance. So if we take into account all of these resistance factors, we kind of have a sum total. And what we end up seeing is that the total amount of resistance that exists in your body, it gives you an idea of how much work the heart has to do in order to get blood to where it's needed. Now, throughout your day, the resistance of your vessels may have to change in order to accommodate blood flow to different regions. For example, if you're running from a bear, you may need to decrease the resistance to your legs in order to get more blood to flow to that area. So how do we do that? Well, if we think about the upper three resistance factors that are most important, we can't exactly, within the 2 seconds you're running from that bear, all of a sudden make the blood thinner. So viscosity is not really an option as far as making adjustments to resistance, at least not very immediate adjustments to resistance. Same with vessel length. We can't just all of a sudden stop sending blood down some vessels if we need to decrease resistance, because of course that means the tissue will die. This is why if we need to adjust resistance and as a result adjust blood flow and blood pressure, we do so through the idea of adjusting this vessel diameter through vasoconstriction and vasodilation. So vasoconstriction and vasodilation ultimately adjust resistance, and this is predominantly done at the arterial level. And this is because we have so many arterioles that we can make very minor adjustments in the hundreds of thousands of arterioles in our body, yet have very dramatic effects. And just to review, if we vasoconstrict, okay, and I'll put this up here, vasoconstriction means we are decreasing the blood vessel diameter, which will increase resistance. And an increased resistance will ultimately lead to an increase in blood pressure. The opposite is true with a vasodilation, okay, where we have an increase in blood vessel diameter. This will lead to a decrease in resistance and a decrease in blood pressure. Now, as you guys have seen, we regulate. How do we regulate blood vessel diameter? Well, we can regulate blood vessel diameter via the nervous system, hormones, but also, most importantly, locally. Now, neural regulation of blood vessel diameter, and since I forgot a slide, I'm going to go ahead and make a white slide here. Okay, so the nervous system regulation of blood vessel. Neural regulation of blood vessel diameters, again, through the sympathetic and parasympathetic nervous system. And actually, for the most part, it is primarily sympathetic. Sympathetic signaling is going to allow for both vasoconstriction and vasodilation. And you might ask, well, how can this occur? It's one signal. Well, with the sympathetic nervous system, what we do is we always see a 50% sympathetic tone. To your blood vessels, which means at rest, your vessels are actually partially constricted. Okay. Now, this means since they're partially constricted, what we can do is we can increase that tone to, let's say, 75%, which would cause increased vasoconstriction. Whereas on the opposite, we could decrease that tone to, say, 25%, which would cause vasodilation. And this is how we neurally regulate blood vessel diameter. Now, we also can hormonally regulate. Let me the screen here because I wrote too big. We can also hormonally regulate. Now, there are three predominant hormones that can all adjust blood vessel diameter. We have ADH, antidiuretic, which is going to cause vasoconstriction. We have angiotensin 2, which is part of the RAA pathway, which will also cause vasoconstriction. And finally, atrial natriuretic peptide, which is our vasodilator. Now, for all of these, antidiuretic vasoconstricts because if you're dehydrated, that means you have low blood pressure, and if you have low blood pressure, we need to increase that pressure rather quickly, so that's why antidiuretic vasoconstricts. Same thing with angiotensin. ANP, on the other hand, is released when you have elevated blood volume. Well, if you have too much blood, we need to dilate to take some of the pressure off while we try to pee out that extra blood. Okay. Now, one thing to remember about both neural and hormonal regulation is they are system body-wide, I should say. So when you release neural signaling or hormonal signaling, it occurs or it results in vasoconstriction or vasodilation everywhere there is a receptor. Okay, but here's the issue though. If we send the signal everywhere, not necessarily all areas need the same level of signaling. For example, with the sympathetic nervous system, great, we've sent the signal everywhere, but if you're running from a bear, we're going to need a slightly different distribution of blood than if we're swimming from a shark. So how do we regulate that? Well, what we're going to see is these two adjust blood on a system-wide or body-wide scale when there's a body-wide issue taking place. But what we can do is rely on local signaling in order to adjust blood flow to areas that are in need the most. And what we see is local signaling always overrides anything from the nervous system or hormones, since a tissue knows what it needs more than, or better than anybody else. So how does this work? Well, tissues require blood flow because they need oxygen. They also need nutrients, they also need other things, but as far as a minute-to-minute, second-to-second need, tissues require constant blood flow in order to maintain oxygen perfusion, because oxygen is needed to make ATP. So what we see happening is increased metabolism of a tissue, results in an increased demand for blood, which means we better increase blood flow to that tissue. And we can do that by triggering vasodilation. Okay? But how can we signal this increased metabolism? Well, what we see is that byproducts of metabolism will trigger changes in blood vessel diameter. So, what are some byproducts of metabolism? Okay, well, how about the buildup of CO2 or the buildup of hydrogen ions, which also equals a decrease in pH? How about an increase in temperature? All of these changes occur when a tissue is doing more work. And if a tissue is doing more work, it needs more oxygen. And this is why all three of these characteristics will ultimately lead to increases in blood flow through vasodilation. Now, something that isn't quite as obvious, since these guys are directly related to metabolism and are often referred to as metabolites, okay, is an increase in extracellular fluid potassium levels will also cause vasodilation. But this isn't quite as intuitive as buildup of CO2. But if extracellular fluid potassium is building up, that means the cell is increasing its action potentials, okay, which means it's being told to do work, right? Because you're not going to send an action potential to a cell, or a cell isn't going to respond with an action potential if it isn't doing work, okay? So the increased number of action potentials means increased workload. Well, if you're doing more work, then you need more O2, and that increased O2 is going to be accommodated with an increase in blood flow and the resulting vasodilation. Here we'll actually demonstrate this in class with one of our experiments. And here we see a little schematic. Sorry, I forgot to change the color of this. Okay, and this is what I had already said. Remember, local needs will always override systemic control, meaning your hormonal or neural signaling. Okay. So what's happening with exercise and TPR? Well, with exercise and TPR, of course, we already saw that we're going to have an increase in cardiac output. But as pressure increases, you might be thinking, well, wait a minute. Drastic increases in pressure aren't supposed to be a good thing, right? Didn't the doctor say I'm supposed to keep my pressure low? But yet he tells me to exercise. Okay? So wait a minute. Not only that, if I'm exercising, then I'm activating the sympathetic nervous system. Doesn't that mean I'm constricting everywhere? Well, don't I need blood when I'm running from a bear? Well, all of these questions come back to the play around or the relationship between the local regulation of blood vessel diameter and in the fact that it will always override the neural signaling. What this allows us to do is that when we're running from a bear, we're going to send out sympathetic signals. And those sympathetic signals are going to be used to direct blood away from non-essential areas. So we're going to get the blood away from the gut, get it away from the kidneys, get it away from the skin for the most part, so that we can only send blood to those areas in need. Well, but how does your brain know what the areas are that are in need? Because remember what I said earlier. The areas in need for a running from a bear scenario are going to be very different than the areas in need for a swimming from a shark scenario. Well, here's the great thing. Only those areas in need are going to have the elevated metabolic demand, which means only those areas in need are going to build up those metabolites, leading to the local vasodilation and the increased blood flow to only those areas or only those critical systems. So this becomes very essential in directing blood to only those areas that are in demand. So for example, in the experiment we're going to be doing in class, the volunteer is going to be biking. So the increased metabolic demand of the muscles in the leg is going to direct the flow to those areas, while vasoconstriction everywhere else serves to conserve blood from those non-essential areas. So this idea of local regulation or auto-regulation, overriding the neural or hormonal regulation becomes very important, because we can rely on the sympathetic nervous system to direct blood flow away from everything non-essential, and then just rely on the buildup of metabolites to get blood flowing to those areas that are actually in need. So very important concept here. And that's at the end of our second lecture. And we're going to kind of hopefully put it all together and have a little bit of an overview of the experiment in our final lecture for part 3.